Good afternoon, everyone. Good afternoon. Welcome to Excellentium2026 in Lyon. First, on behalf of Global Biotech Revolution, I would like to thank you. Thank you all for making the travel to come here today. I know it has not been easy for everyone. Some come from very far away countries. So really seeing this auditorium with 100 leaders of tomorrow coming from more than 40 countries, it's really an honor for us to welcome you today, to host you today. So thank you. This would have not been possible without the support of our generous sponsors and partners. So I would like to properly thank our partners, so Amgen, Sanofi, Heats Institute, Qiagen, Thermo Fisher Scientific, Lundbeck, ARSK, Institut Mérieux, Syngenta, Lundbeck, Mad Pharma, and Only Lyon Invest. And also I would like to thank the EM Lyon Business School, which is a global exceptional school that I actually also attended, to open their doors to us, to all of you, and to our community. So just before we move on, I'd like to give, so we can share together some logistics aspects. So I'll let Nikki lead those. Thank you.
Hi, everyone. Really nice to meet you. Welcome and congratulations on being here. It's truly an honor, as Dan said, to be here with all of you. Before we get into the fantastic speakers we have planned for this afternoon, just a few minutes of logistics, very, very important. So please do follow along for these. As you have read, sort of all the sessions are mandatory to attend, and it would be great if you could attend 5 to 10 minutes earlier to make sure that there are no interruptions during the sessions and ensure that all of your phones are on Do Not Disturb or silent. Please be respectful. Please do ask questions, but also please allow all your other fellow LOTs to have the opportunity to ask questions as well. So if you have asked one question in a session, please consider saving it towards the end so that other people have a chance to. As you all know, our accommodation is in Novotel. Some of you might have already been there. Sort of the check-in, check-out times are here, and sort of I'm sure you've already been dealing with this, but if you have any questions, please reach out to me or Camille on any of those aspects. Meals, breakfast will be served in the hotel, very lovely breakfast, and all your lunches will be served in Coeur Baton, which is just outside. Hopefully you've seen that as well. And then all of your evening events and all of the details, all the speakers' information, LOT details, all of that can be found on Notion, which is also accessible through your card here. And then finally, some miscellaneous reminders. You all probably have seen it's very hot, and it will only get hotter. So please, please make sure to hydrate, keep water with you at all times, have a hat, have sunscreen, stay in the shade if you feel the need to. And then finally, the two maybe most important things, wear your badges at all times so that we can identify you. And please make sure to keep notifications on for Slack.
So now to begin this event, I'd like to handle it over to two exceptional speakers. So to introduce them briefly, most of you, I guess, have met them already during the tour. They are two of the people who know the bio district in Lyon better than, I believe, anyone. So they are absolutely amazing, and I hope through the tour you had the chance to have a glimpse of what will happen and what you'll discover during this program. So the first speaker is Bruno Versailles. He is the head of the HITS Institute, HITS for Healthcare, Innovation, Technology and Society Institute. He's also an amazing teacher in economics from this school. He has published in many well-ranked and very famous publications like the Journal of Health Economics and others. He also has supported many biotechs and life sciences companies worldwide, and of course he has been key for this event to happen. He is a key sponsor for the event. He really supported us for a whole year now, and without him this would have not been possible. So I would like to take a moment to really warmly thank you, Bruno, for making this happen. So, and alongside him is Florent Gerbeau. Florent Gerbeau is also sponsoring the event and partnering with us and supporting this event. He is the lead advisor in life science of Only Lyon Invest. He really makes sure that all the international startups in biotech, life science, medtech really are able to set here in Lyon to find their place, to develop strong strategic partnerships, find about funding opportunities and really supporting them in this ecosystem. So thank you, Florent, and I believe with those two exceptional speakers, you'll be able to understand why Lyon, why this biodistrict is really a very strong biodistrict, health biodistrict in Europe. And I would like to ask you to join me in welcoming them very warmly. Thank you.
Can you hear me well? Yeah. So welcome everyone. It's such a pleasure to be here with you. Okay, it works. Congratulations for making it to Excellentium. I know that your applications have been going through a very selective process, and this explains why we have such impressive individual profiles here in this room. Obviously, it's for me wonderful to see you all here. So for that reason, my turn. I would like to thank warmly, very warmly, the outstanding team of Global Biotech Revolution, starting from Dan. Please, a round of applause. And Camille. And the whole team. I don't know you very well yet, but we have time to start conversations over the next 3 days. As a director of the HITS Institute, if you don't mind, I would like to share with you some words about who we are. And the first thing is that we are located, and you are located now, in the part of France where the biopharma industry footprint is the largest. And for that reason, we at IM Lyon Business School, we are positioning ourselves as the French business school of reference in that domain. Florent will say more about that later. Our activities span education, research, and outreach. We offer a range of graduate programs, including this one, which is MSc in Healthcare Innovation and Data Science. It attracts highly qualified students from everywhere each year. Pass the word if some of your friends are interested. Probably it's a little bit too late for you because you have reached a higher level in most cases, but I know you would be wonderful ambassadors. We also design executive education programs that we tailor-made for pharma companies or for other healthcare-related organizations, like networks of labs or private and public hospitals. You might, over the next 3 days, bump into some of our researchers. They are interested in a range of topics that are at the crossroads of healthcare and business, obviously. And we also are happy to host academic visitors like Tongshi Kai a few months ago from the US. The HITS Institute supports the incubation and acceleration of startups. There are many around us, and some of them might be familiar to you. I know that you will meet Hervé Afaga. I think it's at day 3. He's co-founder and CEO of one of these startups, the name of which is Mat Pharma, and he's also an alumni. We also organize conferences. Last year, the topic was the use of AI for the discovery and development of drugs, vaccines. We are in Lyon. Biomarkers and diagnostic tools. And like for Excellentium, we had an exceptional lineup of speakers, including, probably you know him, Alex Zavoronkov, who is a co-founder and CEO of In Silico Medicine, which is one of the frontrunners, or the frontrunner actually. We also host visitors, personalities who are interested in starting conversations with our students, like Frédéric Oudéa, who is the president of the board of directors at Sanofi. We have academic partners. One of them, an important one, is the UCL Global Business School for Health. Recently, we signed a dual degree agreement in biotech and pharmaceutical management. We also take part in international events, like the One Health Summit that was organized recently in April by the French presidency. So we contributed to it with our partner, Institut Mérieux. The same month, it was a very busy time, we also co-organized with the Harvard School of Public Health the French hub of a global hackathon. And the winning team here made it to Boston because they were selected among a very large number of projects. In my recollection, 1,553, they were in the 30 finalists, so they were invited to Boston. And in case you are interested in returning to Lyon shortly, in a few months from now, we will be hosting the most important conference on infectious diseases in France, which is organized by Map Design, and of course you are very welcome. So over the next 3 days or so, you are going to learn, you are going to share, you are going to connect, and hopefully at the end of the process, at the end of this week, you will be leaving Lyon with new perspectives in mind, fresh ideas, and great friends, and be certain that we will do our best for this to happen. Thank you for your attention.
Good afternoon, ladies and gentlemen. Dan, thank you very much for your warm welcoming words. We are truly delighted and very proud to welcome you and Excellentium to Lyon. And we suppose that it's not only by chance that you come to Lyon, or because we have an excellent and internationally renowned business school with the Lyon, but because Lyon is also one of the European major hubs in life sciences. And I would like now to give you a brief overview about the richness and the density of and the dynamic of this hub. Some of you have taken part in the walking tour in the Biodistrict Gerland and have got a first glimpse on it. Now let's step back a little bit and have a look at the bigger pictures. As you are now in Lyon, you all know why it's Lyon, but however, I will start with a short geographical reminder. Lyon, it's a 2 million inhabitants metropolitan area. We are located right in the middle of France and Western Europe. We are 2 hours from Paris by high-speed train, with a train every 30 minutes. We are 1.5 hour from Marseille and Geneva, 4 hours from Milan, 5 hours from Barcelona, 4 hours from Brussels also. And by plane, we are within 2 hours from almost everywhere in Europe. Lyon is today a major hub in life sciences with deep historic roots, and interestingly, these roots are linked to the silk industry. And Lyon was in Renaissance the Europe capital for silk, and producing silk requires 2 capabilities: chemicals, chemistry, to dye the fabrics and to make the fabric softener, and mechanics to wave them. And from the chemicals, we have got the chemistry industry. As you've seen in the video, we have also large chemical industry. And this industry, this chemical industry, evolved to the pharmaceutical industry and to the biotech industry. And from the mechanics, we have got engineering expertise, which led to leading medtech sectors, and also automotive industry, but it's another story. Today, Lyon is the first manufacturing region for vaccine in Europe, and also one of the major hub for infectious diseases in the world, with companies like Sanofi Vaccines, Boehringer Ingelheim, bioMérieux. And these 3 companies actually are linked to the story of the Mérieux family, which legacy remains very important for the city. I think you will have a talk from Alexandre Mérieux today or Wednesday, tomorrow. And Alexandre Mérieux is the CEO of bioMérieux. He's a force in the story of the family. And if we look at the great-grandfather from Alexandre, Marcel Mérieux, he was a student of Louis Pasteur and one of the first in the world to industrialize the manufacturing of sera and then vaccines. And he did that in Lyon at the end of the 19th century. Today, as we still have bioMérieux. bioMérieux is a global leader in vitro diagnostic. It's headquartered in Lyon. They have a big R&D facility, big manufacturing facility in Lyon and outside of Lyon. Sanofi Vaccine also originated from the Mérieux group. Sanofi today they operate 4 R&D and manufacturing sites in Lyon, focusing on vaccines, immunotherapies, and its mRNA Center of Excellence. And then the same applies to Boehringer Ingelheim. Boehringer Ingelheim, it's a German company. They acquired 6 or 7 years ago a company called Merial. Merial was the veterinary branch of Sanofi, and they operate in Lyon manufacturing site for veterinary vaccine, and a large research facility on animal health. The region of Lyon is also the leading region in France for manufacturing for biologics and manufacturing of API, active pharmaceutical ingredients, with companies like Sequence. Sequence is headquartered in Lyon. Explora is a joint venture between a German company, a French company, and I think it's a Spanish company, the third one. We also have Siegfried, is a Swiss company here, and Euro API. And a part of, though I should also mention, even if it's not the main topic of Excellentium, we are also the leading medtech industry, medtech region in France for manufacturing and subcontracting in front of Paris. Mostly we are always the second after Paris, but in this case we are the first. We also have headquarters of company, a part of bioMérieux, Sanofi Vaccine, and Boehringer Ingelheim. We have Viatris in Lyon. We have Merck Group, and other pharmaceutical company like Agita, which is a company from Lyon. Aurobindo, we have an affiliate from Aurobindo, the Indian generics company, and so on. This dynamic ecosystem is not only bringing together big pharma companies, but also around 800 startups and SMEs, not only in Lyon, in the region of Lyon, saying from Clermont-Ferrand up to Grenoble. And also 800 SMEs and startups developing new products and technologies, but also numerous CROs, CDMOs, CMOs. Actually, a biotech company wanting to develop a product in the region of Lyon can find almost everything from the drug discovery part up to the manufacturing. Roughly half of this company are in the biotech and pharmaceutical sector, and the other half in the medtech and digital sector. If you look at the therapeutic areas, we have in Lyon a strong specialization in infectious diseases coming from the Mérieux family, from this history. And this accounts for about 25% of the companies that we have. This is quite unique in Europe and maybe in the world. Other areas of expertise are in the neurosciences, as I mentioned before as we talked. There's a lot of company and a lot of academic research in neurosciences. Dermatology and regenerative medicine also, and oncology, of course, but a little bit like everywhere. This industrial strength is supported by world-class research with several internationally renowned research centers. In infectious diseases, as already mentioned, you will learn a lot about infectious diseases in Lyon with the series International Research on Infectious Diseases that we saw in the Biodistrict. As a part of the institute is in the Biodistrict. Vet AgroSup is a vet school also. By the way, we have the oldest vet school in the world. It has been founded in Lyon in 1761, at the end of the 18th century. They are a little bit outside of Lyon. We have also research institute in oncology, of course. The CRCI Lyon Research Center on Cancer is linked to the Comprehensive Cancer Hospital Centre-Léon-Bérard. Centre-Léon-Bérard, if you know, is one of the major European comprehensive cancer hospitals. It's like in Paris, Gustave Roussy. Mostly people know Gustave Roussy. But in terms of patients, it's smaller than Gustave Roussy. But in terms of research, with the CRCL, we have the largest research center on cancer in France. Yeah. Neurosciences, 4 different research centers, and a huge hub of neuroscientists and clinicians, around 700 neuroscientists in the region, in one hub, actually at one place, and 300 neuroclinicians. Expertise also in tissue and skin engineering, functional genomics, cardiovascular and metabolic diseases, and also therapeutic application of ultrasounds. That's also a specificity of Lyon with different startups coming from this institute. In terms of clinical research, we have major assets. As Centre-Léon-Bérard is one for cancer, but also the STL, the University Hospital, second largest in France after Paris. Actually, it's a network of 13 different hospitals in the city, 26,000 people working for them, 5,000 physicians, and more than 2,000 active clinical trials. As a whole, Lyon offers access to a large and highly qualified pool of talent for companies wanting to come in the region of Lyon. These different assets in the pharmaceutical area and biotech area are concentrated in some hotspots in the city. We, for some of them, have already made a tour in the Biodistrict, which is in the south of the city. As we just visited half of the biodistrict. The biodistrict is around 5,000 people working in the life sciences here. 70 major players, research labs. I mentioned the CIRI, but the LBTI in tissue engineering is also there. The BSL4 lab, we have a BSL4 lab, the largest BSL4 lab in Europe, civilian, and a lot of biotech and pharma companies, and two World Health organizations are in biodistricts, the WHO Academy, and the International Agency on Research on Cancer, where you will be tomorrow, I think. Another hotspot is a biopark in the Eastern hospitals. We have there around 30 biotech companies in dedicated building with lab space. 3 hospitals around them, the Edouard Herriot, Neuro Hospital, Cardio Hospital, Mother and Child Hospital, the Comprehensive Cancer Hospital, Centre Lambera, and also a psychiatry university hospital, very large one, and School of Medicine, Pharmacy and Nursing also. And then in the north of Lyon, the campus Lyon Tech Ladoua with 20,000 students, not only in biology but in chemistry, artificial intelligence, engineering sciences, a lot of researchers, a lot of PhD students, and we have an incubator for life sciences and tech there also. That's the biodistrict, so I won't go a little more further in it. We already visited half of the biodistrict, as you can see. So you have big pharma companies, Sanofi, BASF, Boehringer Ingelheim, Viatris, a lot of biotech companies also, research institute on infectious diseases, tissue engineering, genomic, and international institution, and French also regulatory agency, like NASM are here. But there is also a lot of companies outside of Lyon. As you can see here, you have in the middle the biodistrict Gerland, but outside of Lyon, one place particularly is very interesting is Marcy-l'Étoile. Marcy-l'Étoile is 30 minutes outside of Lyon in the west. In Marcy-l'Étoile, you have actually the headquarters of bioMérieux is here, is located here. And you have so R&D here, you have manufacturing facilities, and Sanofi is also located here with the largest vaccine manufacturing facility, I think, in the world. Located here for pediatric vaccine mostly, and then the vet school is also here. Marcy-l'Étoile is 12,000 people commuting every day to work in this small city. In the south of Lyon, some CDMO, CMO, and manufacturer of APIs like Sequence, Explora, I mentioned before, Patheon, Thermo Fisher, they have big facilities outside of Lyon. Sky Pharma is a French CDMO. Or Bentalion. Bentalion is a French-Lebanese company. That we bought, INT, we bought this company to Lyon. And in the west, you also have the Boehringer and or medtech company like Stryker, Baxter, Merck. So I hope you will now have a better overview over where you are, where you will spend these 4 days. Part of the speaker you will have come from Lyon. You will have speaker from Sanofi, from Mat Pharma, it's a company working in microbiota, in oncology, from Adocia, I think, and Osivax, developing vaccines. So enjoy your time in Lyon, and I will be here today, this evening, and tomorrow. If you have any questions, don't hesitate. Thank you very much.
Hello. And oh yeah, great. Okay, and now it is my great pleasure to welcome Dr. Andrew Singleton. Dr. Andrew is one of the leading experts in the field of neurogenetics, and his work revolves around understanding the genetics that goes behind neurodegenerative diseases, especially Parkinson's disease. And He is leading the global Parkinson's genetics program. And one of the amazing things about this project is that it has studied over 150,000 DNA sequences of a very diverse set of group of people. This gives us a better hope and idea of understanding Parkinson's disease better in the future, and also to be able to develop further therapies. And Dr. Singleton, we are very fortunate to have you today in Lyon at Excellentium 2026. And your field of expertise is something that we really appreciate. Please have a warm welcome and a round of applause for Dr. Singleton.
Great, thank you so much. So I asked Google Gemini to create a picture of me that made me look approachable yet professional. I'm not sure that it's worked, but yeah, that is not really me. That's what AI thinks I look like. So hi everyone, my name's Andy Singleton. For those of you who I haven't met, which is probably most of you, I worked in the federal government, in the US government, for about 26, 27 years, running a lab that focused on understanding the basis of neurodegenerative diseases. So we did a lot of work in Parkinson's, also Alzheimer's and other related diseases. I'm a geneticist, really. That's my core expertise. So I actually don't really think of myself as a scientist in many ways. I think the way that geneticists think and the way that we kind of operate at scale, I think of myself a bit more as an engineer or someone who's really interested in operational things, and you'll get a flavor for that as I go through the talk that I have planned here. I have to say, please, if there's something I'm talking about that doesn't make sense or is really boring and you'd like me to move on, shout out, ask questions. It will be far more interesting if there's some back and forth. So as I said, I worked for the federal government until about a year or so ago, but whilst I was there, I did a whole host of things. I had started my own group, then I took over a department, and then I launched a new center for Alzheimer's disease. Those things were all really good fun. I really enjoyed them. I really enjoyed building things. But this project that I'm going to talk to you about today is one that I just love. I love this project. I've been working in the field of Parkinson's genetics for many years, but this was a project that allowed us to think in a bigger and broader scope and scale. So I run this program with a guy called Cornelis Blauwendraut. He's a Dutch guy. The program started in 2020, end of 2019, 2020. So we were starting a project that aimed to understand the genetic basis of disease in populations all around the world, and then 3 months later, the global pandemic hit. Now, I will say what this allowed us to do is really spend a couple of years thinking really carefully about operational challenges. And as you'll see from the scale of the project, there are quite a lot of operational challenges. So we're very, very lucky in that we're supported by a single funder. So this is philanthropic funding. This funder is the ASAP program, the Aligning Sites Across Parkinson's program, and they fund a whole bunch of things. They fund principal investigator-led grants, so that's the Collaborative Research Network, and then they fund a whole bunch of what you can think of as resource projects. So these are projects that are aimed to set up to address a problem where we kind of know what we need to do, and it's just about establishing scale and moving forward. So GP2 was the first resource project. I'm involved in a couple of the other resource projects, including the Indie one, which was aimed at creating stem cell lines for about 800 different neurodegenerative mutations. So GP2, the Global Parkinson's Genetics Program, we have a very, very simple goal, and that goal was driven by a couple of things. First of all, we made really good progress in understanding the genetic basis of neurodegenerative disease. Lots and lots of progress since early 2000. The problem with that progress, though, is that it's made almost entirely in people of Northern European ancestry. So this is an issue, right? If we think about precision medicine, if we think about this notion that we'll start to come up with targets that we need to apply to a person based on the underlying mechanism of their disease, then of course we need to understand mechanism in everybody, not just a subset of individuals. So the real driver for this work was to create a global community of researchers who could accelerate our understanding of the genetic basis of Parkinson's disease. In the past, when geneticists, particularly from well-funded labs in North America and Europe, when they were interested in looking at underrepresented populations, generally what they would do is jet in, grab a bunch of samples, work with a local clinician, take those samples away, and then kind of the next time that the clinician, the local group that was working on this would hear about it is when a paper's being written and they're being asked for their viewpoint. What we wanted to do is something different. We wanted to create a global research community. So we really wanted to enable research in groups all around the world. So think about that as I go through what I'm going to talk about today. Failure for us would be if my lab and a couple of other labs were just churning out papers on these data. Success is if we can create this engine of researchers all around the world who now have access to the data and the skill sets required to analyze the data in order to be able to move forward. In terms of the resource we'll produce, so when we started coming up with this idea in 2019, we thought 150,000 people around the world. We'll be able to get DNA on 150,000. And I wasn't sure that we could. You know, that seemed like a lot. No one really knew where all the patients were or whether the cohort providers would want to collaborate. I would say at this point we have surpassed that. We're past 150,000. I think I have here 250,000. I think we'll end up with more than that. So we're generating deep genetic data and harmonized clinical data on somewhere in the region of 250 to 350,000 people with Parkinson's and related disorders. And so the output is the one that I've already told you about, creating data, creating skill sets to create an engine that is just driving discovery. Not one that where we're pushing out manuscripts, but one where the consortium, the collaboration is driving progress. So how have we done this? Let me run, what I thought I would do is go through how we've set this up, give you some vignettes of where we are and what we're establishing. I'll talk a little bit about where we're going over the next few years, and then maybe some lessons learned at the end. So the first thing is we are now working with cohorts in 76 countries. I think there are around 400 cohorts, and have focused really hard at building collaboration, building trust, and building capacity for local researchers. So what does this mean? So part of building capacity is building skill sets and talking to researchers around the world and understanding what they have capacity to do, what they don't have capacity to do, right? So in some countries, there's already really good computational expertise. In some countries, there is really good biological expertise for sample processing. In some, there's no stable electricity, so you have to kind of think about, well, what can we do to help those folks also take part in research? So a large focus of our work has been on training. You can go to our website. We have a learning management system, and you can learn how to go from not really knowing how to do any genetics all the way through to doing very complex genetic analyses. We have around 100 or so videos translated into 100 different languages. They're all kind of free for people to access. I think we have around 1,500 users within our network now who go and access those things and basically learn how to do genetics. About a third of the people that use these courses are from low and middle income countries. We've also built some physical resources. So we want to build capacity, sustainable capacity in some of the places that we work. So about 3 or 4 years ago, we contributed towards building a biobank in Lima. Leon's on my mind, in Lima, with the notion that this group would then be able to support sample processing, sample extraction, and some genetic analysis for a future after we're gone, right? So creating skill sets and creating physical resources. And we just have finished building a biobank in Lagos, Nigeria, again with the same idea. There's a really great group there. Can we enable them to do research beyond just what we're doing in GP2? We sponsor a lot of students. So I think we have now 30 or so PhD students. And the requirement to be a PhD student in GP2 is generally that you come from a population that is underrepresented in research, so from an area that's underrepresented in research. And we look for two things mainly in those students. They go through a whole round of interviews, and we're looking for them to have a commitment to staying in their local community and enabling education and enabling further training and growing the skill set in their local community. And for that reason, we register these individuals at local universities all around the world. They're working on a whole bunch of different things. We also have some online master's courses, so teaching people data science from a US university, but they can be anywhere in the world doing that. The other piece that has been really key, I think, and I think this is something that my boss, I worked for a guy called John Hardy for many years. My boss taught me very early on, which is to work with groups, there's nothing like going to meet with people and build trust face to face. So we do a lot of traveling. I spend about half of my time traveling, but we also hold annual meetings. So in the even years, we hold regional investigators meetings. So we go to different regions and bring people into those regions to learn about what we're doing in GP2. And then on odd years, we have one central meeting. So these meetings you can see in red have all happened. They all have somewhere between 150 and 250 people that we fly in, and we spend 2 days talking to them about the consortium. The green ones, I'll be going to Montevideo in a month's time, Seoul a couple of months after that, Amsterdam early next year. So thousands and thousands of interactions, as you're doing here, where you start to build networks, you start to build trust, you start to get people working together. Again, the idea here is to build a network. This component is a piece that I've really, really enjoyed, which is, you can imagine that organizationally this is a lot of work. And we have to have quite a complex organizational structure and lots of different things going on. One of the most fun things that we did early on was that, of course, we brought in very established researchers in genetics and Parkinson's disease, people that had been around for a long time. But for every leadership role that one of those folks fits, we pair them with a new researcher, usually a younger researcher, but someone who's new to the field, so that they could learn from each other. This was incredibly helpful for the senior folks who were sometimes too busy to look at the details, and incredibly useful for the junior folks who got to learn about how do I run meetings with 30 different people who speak different languages and keep people on track and make sure that we're delivering what we need to deliver. So growing this next generation of leaders has been key. This is a small subset of all the working groups and interest groups that we have. Every one of those is headed by a senior leader and a junior leader, and they work side by side. One is not in charge of the other. And this has been really fun because you've got to try and find really good, talented people at the beginning of their career and give them opportunities to grow over time. That's been a lot of fun. Accelerating discovery by democratizing data. Something we've been really, really good at in the genetics field is sharing data for years. We're really good at putting up genotypes onto a website that you can log into and access. But that's not the same as making data available, really. It's just saying it's there. Really what was happening was the same old labs were analyzing the data because they were the only ones who had the skill sets to analyze the data. And they were the only ones who had the computational resources to analyze the data. So what we've tried to do is break that mold a little bit. So the way that GP2 works is that we reach out to cohorts people who have collected patients or people who are interested in collecting patients. We work with them through a whole series of legal things, and eventually DNA or blood or saliva or something like that comes into GP2. Clinical data comes into GP2. We generate genetic data. We generate and harmonize clinical data. And then the first people to get that back are the cohort contributors. And they own that data. They can do what they want with that data. But they are also then a part of GP2, and they get to access every other data set that's in GP2. So all of our data goes to a central cloud-based platform where anyone within the network can access it immediately, and then a short time after that, anyone in the world who's a bona fide researcher can access that. So the nice thing about this network is it's a distributed cloud-based network. So it allows us to deal with all of the legal issues around where data can be and where it can't be. Our European data stays in Europe, our American data stays in America, Indian data in India, et cetera, et cetera. But it also means that we can provide compute. So you don't need your own supercompute cluster. We'll pay for it. We'll pay for you to use it. And we've previously provided the training for you to use it. So it's really about democratizing access to data, not just making data available, giving you the skill sets to access the data, and giving you the research capacity to access that data. One component of this is teaching people one-on-one or face-to-face data analysis. So we have a group of 3 or 4 people who travel around the world, and they go to various sites and do a 2 or 3 day workshop on advanced analytics. So they'll go to, so this one in Malmo. Went to Malmo, sat down with a dozen or so trainees. During those 3 days, the people that are running the course are looking for 2 or 3 trainees who really excel. So the next time we go back to Malmo, our group co-runs the course with those 2 or 3 really talented students from the first course. The third time, those 2 or 3 talented students run the course by themselves, and we help online. So we have this train the trainer model. I think we've done 20 or so of these now, as you can see in North Africa, South America, Southeast and Central Asia. So these have been great, I think. Our data is all available. If you want to have a look at the data, you can access it through these QR codes. So the last bit is about transformative genetic methods, and actually genetics is it's a product now, really. It's when I was running a lab back in 2006, 2007, you really wanted to generate the genetic data in your own lab. And you just don't need to do that now. It's so standardized. You just pay someone to do it, send it out to a company, have it generated. So we have produced a lot of genotyping data. So we use a 2 million SNP array, a whole lot of whole genome sequence, so sequencing the whole genome, and also some long read sequencing. So this is where we are in terms of data. We have just released data on about 140,000 unique individuals. So I'm showing here just the genotype data. So 125,000 people with genotypes, about 65,000 with whole genome seq, and about 50,000 with extended, really pretty extended clinical data. So you can log on to the GP2 site. You can access this data today. It's relatively straightforward to access. We do data releases twice a year. So each data release, we add about another 30,000 people, something like that. We have pretty good representation. If I had shown this map 6 years ago when we started this endeavor, there would have been a couple of dots in Europe and a couple of dots in North America, and that would be it. But you can see that we've really launched a lot of studies in other places around the world, which has been enormous fun. It's been really great fun to see the talented folks in those spaces really take advantage of this opportunity and grow. Obviously, there are places we still need to expand into, and we're working hard at that. So that's all great, but what does it mean? You're generating all this data, you're teaching people how to use the data, you're growing the next generation of leaders. What does it all mean? So again, it comes back to this notion that I've mentioned a couple of times before. We're trying to create the next, a self-propagating engine of discovery. So a group that, if I wasn't here tomorrow, would continue to generate results, generate new insights into disease. And the good thing is that we're there now. We're at a point now, we've reached an inflection point in the last year or so where the engine is running really smoothly, data's pouring out, and we have a lot of people analyzing the data. So I'm going to give you just some small examples of the types of work that are being done in GP2 now. This by no means represents the scale of what we're doing, but I'll give you some examples. So the first example is one that is based on scale. So we're able to collapse enormous amounts of data together to do large analyses. This is actually in European ancestry individuals, but we're able to bring together data on 60,000 people with disease, about 1.5 million people without, and really, really dramatically accelerate genetic discovery. So here we have 140 risk loci, novel risk loci for disease. Each one of these is giving us a window into the etiology. Each one of these is telling us about what's a good target, what's not a potentially good target. It also starts to give us ideas. We can overlay lots of other omic data, and I'll come back to this, to give us ideas about, well, okay, you know the gene, or you know the location of the gene. Can it tell us something about the cellular context or the effect of the gene? Is it affecting transcription? Is it involved in mitophagy? Is it involved in some other process? So we're able to now start to bring large data sets together to get insights into this space. We're also able to look quite broadly. So one of the huge challenges in our field and in many other fields is that we all believe in this notion of precision medicine, of this idea that if you have a mechanism and you understand a mechanism that's involved in disease, targeting that mechanism in people where you know that mechanism is occurring is particularly important. So this creates a really big challenge, because often in this early days of precision medicine, when we say we're targeting a mechanism, what we're saying is we're targeting a group of individuals with a particular mutation, because we don't know how to subdivide up others without mutations into mechanistic subgroups yet. The challenge here is that we only know about the genetic drivers in a couple of populations, and we only tend to run trials in a couple of populations. So if you look at the kind of pointy things with dots over them, this is where trials, precision trials for Parkinson's disease are occurring. And you can see overwhelmingly they're in Europe, they're in North America, a couple in Southeast Asia. But if you look at the distribution of mutations that are being targeted by those trials, they're dramatically different, and there are populations where these mutations are incredibly prevalent. I'll tell you a story in a minute about a particular mutation in Africa that's remarkably prevalent. So we need to understand more about the distribution of mutations and the distribution of causes of disease, and then try and enable groups to run trials in places outside of where they would ordinarily go. So this is mutation data for about 100,000 people around the world. And I should explain. So GBA is a gene that contains mutations that cause Parkinson's disease, LRRK2 the same, synuclein the same, etc., etc., etc. Diversity driving discovery, right? There are two compelling reasons to understand the genetic basis of disease in ancestrally diverse groups. The first is parity. If we are to treat a global disease, we should understand disease in all populations so that every population will have access to targeted therapeutics as they happen. There is another driving reason, and that's a scientific one, which is that we can combine data across populations to give us much more knowledge than we would ever get by looking at one population. It allows us to fine map signals. It allows us to really get ideas about what the gene is, what the effector is. And this is a very early example of that kind of work. So rather than looking at the genetic basis of disease in one population, we're looking at it in lots of different populations and looking for things that are unique, things that overlap, things that are different. Sometimes they have different effects in different populations. So this is a very early study from GP2 showing that just by combining very small numbers in this instance of novel populations, we can identify new risk factors for disease. We're also able to focus on individual populations. So I talked a little bit about training. When you do a genome-wide association study, what you're doing essentially is looking at genetic variability in lots of people with a trait, in this instance disease, and comparing them to lots of people without that trait. And usually to find something, you have to look in thousands of individuals. It's just because of the statistical power required. We were working with a group in Lagos, Nigeria, in West Africa, run by an amazing clinician called Njideka Okubudeju. And she had collected around 1,000 cases of West Africans with Parkinson's disease. So we were teaching two groups how to analyze this data. So 1,000 cases, 1,000 controls, something like that. You never, ever expect to find something in that small a series. So my group, which was at the NIH at the time, was teaching a group in London and a group in Lagos, using our web-based platform, so you can do everything asynchronously. You know, we can run something when they get up in the morning, they can look at the results, etc. And amazingly, we had this hit. So for those of you who don't know these plots, these are called Manhattan plots, and the higher the dots, each dot is a genetic variant. The higher the dot, the more significantly associated with disease the variant is. So here there's a variant that's associated with disease at a p-value of 10 to the minus 14. So really, really significant. You would never, ever have expected to see this. We never would have done it unless we were doing it as a training exercise. What it turned out to be was a variant in a gene that we knew was associated with Parkinson's disease, but a new type of variant that affects gene splicing. At first blush, it looks like it increases the expression of the gene, but it doesn't. It increases expression of a faulty form of the gene, so effectively it leads to a decrease in protein and a decrease in the enzymatic activity of this particular protein. So immediately, this was a new mechanism. The other component that was incredibly striking is that 50% of Parkinson's cases in West Africa carry this variant. That's an enormous percentage, one that we would never have expected. So you have a new mechanism in a known gene, a readout for that mechanism, which is decreased protein levels and decreased GK's activity, and now a population that would never have been included in a precision trial that's completely opened up now for a precision trial. And there are several phase 2 trials for targeting GBA right now. So this would never have been tried in the African ancestry or African American groups, and now those have been opened up to this type of work. And then, of course, we do more and more work in these populations and find additional things. So this is also a new finding in the West African population, a particular LRRK2 mutation that is common, about 2 or 3%. Again, this is important because companies are desperately looking for LRRK2 patients to run their trials. In fact, a trial just failed or just was stopped about 3 or 4 months ago because there weren't enough LRRK2 patients. So finding new patients in different populations is absolutely key. And then lastly, you can imagine that we're doing all these genetic analysis on, okay, is this a risk factor for disease? But you can do tons of other things. Is this a risk factor for comorbidities in disease? Is this a risk? Are there genetic risk factors for response to treatment, for pathology, for all sorts of things? So I show this as one example of this kind of sub-dissection, where we look at genetic risk in men versus women. And by and large, the genetic risk for men with Parkinson's disease and women with Parkinson's disease is pretty much identical, except for 3 or 4 things. one of which I show here. So on the top, you see p-values for association in women. On the bottom, p-values for association with men. So this is a locus that we had thought, a genetic region that we had thought for many years was just a general risk factor for Parkinson's disease. But it's just a risk factor in women. I don't know what that means, but biologically, it's got to be really worth investigating. Incredibly interesting stuff. So what about impact? This is a plot of genetic discovery in PD. 2020, GP2 came along, and you can see that we've had pretty big impact on discovery in Europeans, but enormous impact on discovery in Southeast Asia, Latinos, Africans, individuals in South Asia. And this will really, really accelerate. A couple of reasons why it'll accelerate. These are the publications from us in the last 18 months. We have 170 active research projects ongoing right now. Part of the way that we've structured GP2 is that every project that's proposed, every project that's being undertaken is published on the web. So anyone who's a member of GP2 can see what's happening, can say, oh, I'd like to take part in this, or hey, I don't think it's fair that you're doing this, and can really get involved. So it's, again, this engine's really, really turning and starting to drive results. So this is what I expect over this next period. Just an incredible acceleration in genetic discovery. That's great. That's the point. This inflection point is exactly what we're hoping for. We are funded through 2030, so the timing feels right. We'll continue to accelerate for the next four years. So this creates an opportunity, though, right? It's not enough for us to make genetic discovery and just put these out in the world and say, okay, here it is, go for it. Because there's a lot more than just knowing what the gene is or knowing roughly where the gene is in order to be able to prioritize this for therapeutic interventions. So I talked a little bit about the funder at the beginning, and one of the neat things about the group that funds us is they reward success and are continually looking for opportunities to speed processes. So we started in 2020, everything was going well. They got us to expand into other diseases. So we also work in dementia with Lewy bodies, progressive supranuclear palsy, multiple system atrophy. They gave us more funds to do more whole genome sequencing, more genotyping, to create and collect more cohorts from underrepresented populations, to start to look in individuals before they get disease, so looking at prodromal at-risk individuals. And most recently, we've been supported to take the next step after genetics. So to go immediately from genetics to trying to make some insights into mechanism or subtyping or other things that are involved in the disease process. So this is what we're focusing on now. Again, the discovery piece, what I've spoken to you about today has been discovery. That's running pretty smoothly. I feel like you can kind of set it and forget it to a certain degree. But we're now supported to do three other things. The first is mechanism. Can we generate data at scale? Again, I think of us as kind of an engineering or an operational group. Can we generate data at scale that allows you to go immediately from knowing that a genetic variant is associated with disease to understanding what it does biologically and where it does it? Does it affect transcription? What's the transcript that it affects? What cell type is that in? Is that in response to a particular stress? What does it do to the protein downstream? Does it affect the metabolome in some certain way? So this first component is around mechanism, understanding biological mechanism. And this involves a bunch of omics, mostly in brain tissue. So this is about de-risking things so that when your new startup comes along, you can come to our data and say, well, you guys have actually done a lot of the data of things that we need that kind of reduce the risk for us to jump on this and carry it forward as a target. The third part is around subtyping. Can we use this genetic information that we're creating to start to divide people into mechanistic subgroups? and importantly, to divide people into mechanistic subgroups temporarily. So I suspect that different mechanisms are going to be important at different stages of disease. Can we use our genetic knowledge and overlay that with all of the assays that I've talked about, but in biologically accessible fluids, so transcriptomics, proteomics, metabolomics, all of the omics, and use that data to say, okay, well, this is the group that would do really well with a LRRK2 inhibitor. This is a group that would do really well with something targeting GBA. This is a group that would do well with those efforts aimed at targeting cellular stress. So this is the third component, subtyping. And then lastly, we know genetics is not the whole story. And what effectively we've done within GP2 is to create a network of people who are really interested in PD. They're not just interested in genetics, they're interested in Parkinson's disease. So now we've deployed a large effort to try and understand the role of environment and environmental risk in disease. Really taking advantage of this global scale. So, takeaways. For me, this is just an incredibly fun project. We have really focused on making discovery on a global scale and really leveraging ancestral diversity to improve our understanding of PD genetics for all populations, right? A rising tide floats all boats, but to level the playing field for those groups that had not really had a lot of work performed on them. To generate mechanistic subtype, mechanistic insight. We have to de-risk these next steps. We're not just competing with other neurodegenerative targets, we're competing with the next oncology target or the next diabetes target. So we need to reduce the activation energy required for a group to come along and say, oh, I think this is a really good thing that we should be targeting as a therapeutic. Matched hand in hand with that is now companies are very nervous. Well, this might be a great target, but what's the population that I need to be looking at? What's my therapeutic population? So trying to create methods to be able to define mechanistic subtypes. And then lastly, this environmental nexus. Can we start to find things that will allow us to reduce risk preventatively for Parkinson's disease by saying, okay, these are environmental things that you should avoid. So I thought the last thing I would say is what I've learned from this, from being involved in this whole process. And I should say that when I started working in genetics, it was when I was working really seriously in genetics, like 99 through 2005, 2006. Genetics was a solo endeavor. It was hyper competitive, and it was small groups all around the world trying to battle with each other. You got enormous prizes if you won, you got absolutely nothing if you lost. Around 2006, 2007, that all changed. And we realized that because the methods had changed, we actually had to work together and work collaboratively. Working in a really super competitive environment is fun, I will say. Those races are fun, but this has been even more fun. Trying to bring people together. And I think there are a few things that I've learned as I've tried to bring people together. The first is that not everyone is motivated by the same thing as you, and actually asking people what they're motivated by. You're surprised a lot of people don't know, but some people do know, and they'll tell you. So it's really good to know what it is that people are motivated by. When you're finding people to put in leadership places in your organization, and for us, we have many tiers of leadership. At the highest level, you've got to get people who completely buy into the mission of what you're doing. And GP2's mission is not an academic mission. It doesn't really reward people academically. So you've got to find academics who say, okay, I'm not too worried about publishing. I'm not too worried about the next grants. I want to make progress. And that's not everybody. We've worked with Trusted Partner. That's been key, the Michael J. Fox Foundation. We've had a funder who bought into our vision. That's key. Someone who says, okay, well, here's money for 10 years, come back in 10 years. That's huge. That also gives you the flexibility to try new things. We don't run any of the work in GP2 like an academic proposition. We run it much more like a company. And that allows us to try new things. new approaches to incentivizing, new approaches in organization, new approaches in how we measure success and how we measure failure. The biggest thing has been building trust, so going and meeting people face to face. Some of the cohorts that we're working with, it's taking me 4 years to get them to commit to taking part, and you just simply have to, demonstrate that you're trustworthy, deliver what you say you're going to deliver, when you say you're going to deliver it, and be as transparent as possible. For this type of work, academics doesn't work. You can't treat this like an academic research program. You have to treat it much, much more like a business with huge focus on mission, but also huge focus on supporting people and giving them very, very clear expectations. So one of the things that I think we struggle with continually is when we're working with an academic collaborator is saying, okay, we assign project managers to everything we do. So we have a team of PhD project managers who oversee every aspect of what we do. And when we assign a project manager to an academic group who's working on a project, it's hard to get the point across to them that when we say, if you tell us you're going to deliver this in 3 months, you need to deliver it in 3 months or the money's going to go away, or we're going to try and find someone else to do this. That's a very different way of thinking about science, and I like it. I think there's definitely a learning curve for folks that we bring on. Now we try and support people, we try and find out why things are failing before they fail. Again, the project managers are key here. I think they're the best thing that we have in our organization. They really allow us to drive forward, focus on our mission, focus on delivering what it is that we're trying to deliver. And with that, I will end. This is a photo of a subset of us that met in Honolulu last year for our annual meeting. So with that, I'll end and thank you.
Thank you so much, Dr. Singleton. Right now, we'll open up the floor to questions. I'm sure there are many. So I think I saw you over there first.
Hello. Thank you for the great talk. I guess the thing I'm very curious about is you have many stakeholders as part of what you're building. Could you give an example of what kind of incentives or buy-in do you have for these different stakeholders, and how do you get them convinced? Because I'm guessing the language or the buy-in that an academic would have would be very different from someone at the clinic or your investors or the charity that funds. So I would love to learn more about how or what language or vision you sell.
Yeah, definitely the language when you describe the study is very different depending on who the stakeholder is. And I think that you have to recognize that you definitely can't please everyone, right? But I can give you some examples. So for the academic groups that we work with, the academic stakeholders, we're generally not interested in publishing, but we're interested in helping others publish. So for them, you know, that's kind of their metric of success, so we can help those groups. The other thing is that we give them the data back. So they're getting millions of dollars worth of genotype data returned to them for free, and we'll help them to analyze it. So we have a core group of analysts who you can call in once a week and, you know, say, hey, what do I do with this? For patients, it's a bit more difficult. One of the things that we have struggled with is returning results to patients. So something patients are really interested in is you know, can you tell me more about the genetic basis of my disease? The challenge there is it is about 10-fold more expensive to produce a clinical data result and return that. So rather than genotyping 250, 300,000 people, we'd only be able to do it for 30,000. We found kind of a halfway there in that we have worked with another group with a study called PD Generation, which has facilitated clinical genetic testing, counseling, and return of results. So we don't do it for everyone, but we've done it for about 20,000, 30,000 people. For our other stakeholders, which are the high-level funders and then the implementation partner. So the high-level funder is basically a philanthropic organization that wants to see progress, and the implementation partner is the Michael J. Fox Foundation, which has lots of groups working on different aspects of Parkinson's disease. So for them, they're really interested in what's the final goal, and how does that push us towards faster cures and better deployment of therapeutics. So we continually have to frame everything that we're doing in that way. We're doing these studies X, Y, and Z in order to enable these companies to take this project forward. So there it's like reporting metrics on biopharma data users, interactions with industry, whether targets are being moved forward or not. So yeah, totally different for each group.
Wonderful talk. Actually, I have two questions. First, allow me to say that, and apologize, I'm a chemist, so kindly correct me if I am wrong. So you showed data about a global scale analysis about genes associated with PD. However, as we all know, association is correlation and doesn't always mean direct causation, unless one performs like an in vivo experiment of knocking out certain genes. So in this aspect, does the program do such a thing? And how do you manage this dynamics of doing the genophenotyping in tandem with an in vivo knockout system to rule out if this gene of interest is doing the PD, the causation? And the second, it might be hard, but hypothetically, how far or near are we in finding a cure for PD?
Yeah, I learned really early on never ever to answer that question. So I'm just going to completely pretend you didn't ask that question. So how do we separate association from causation? So it's difficult, but it is also difficult to envisage a situation where a genetic variant is associated with disease and unrelated to the disease process. It can happen for sure, but it doesn't seem to happen very often, at least in my experience. Part of that is doing what you said, right? So looking for functional connections. Part of it is looking in different populations, right? Again, that would reduce your confidence that it's causation rather than association rather than causation. I think the other piece is that which is something I'm really, really excited about at the moment, which is that we are now starting to find biomarkers associated with disease that are sometimes predictive of disease or early stage disease. And I think once you start to overlay genetic risk that we're finding on when those biomarker changes occur, then you start to really place genetic variability into a temporal context of risk. And there, I think it would be really hard to think about association, not causation. Does that make sense? Good.
Okay, we might have time for one more question. So I guess.
Yeah. My name is Noor, and speaking as a data scientist, right? Like I see that you're generating like loads of data. So that for me, like as a data scientist, like crude oil, you know, to take that and play around with it and maybe like train machine learning models. But was thinking I'm curious about the kind of use cases probably that's been used already for machine learning, personalized medicine, diagnostics, you know, using these genomic data.
Yeah, I'll give you a use case that's kind of hot at the moment. So there are a couple of fairly well-funded trials targeting LRRK2, which is a gene that contains mutations that not really cause, but really, really increase risk for disease. Those mutations are relatively rare. They're only present in about 2% of the Parkinson's population. So you can imagine if you have this therapeutic that is focused on modulating LRRK2 activity, the best population you could do this in are those folks who have a mutation in LRRK2, right? It really matches the therapeutic strategy with the mechanism in the patient. However, trials are failing because there aren't enough patients to recruit into those trials. So a use case that the data is being used for now is, can we use genetics and other biologics to define LRRK2-like individuals in the general population? So those people who don't carry a LRRK2 mutation, but the same mechanism is driving their disease. So one approach that's being used for this is to look at omic readouts from accessible tissues in LRRK2 cases, and to try and find a readout that is then present in a subset of PD patients, and say those are the patients we should be putting into LRRK2 trials. So that's one example.
Thank you so much.
Yeah. You're welcome.
Let's give another round of applause for Dr. Singleton. Thank you for being here with us today. So we now invite everyone outside to the common area where you were waiting before the session began for a photo. And then after that, we will go to La Table for our drinks reception. Thank you. So please follow through those doors outside to the common area, and we will take the group photo. And please do not leave anything in this room. You will not have access after 6 p.m., so please make sure to take all your things with you.
Thank you.